test this out?
Yes,
Get it good for a trial, make sure it's good.
good.
Well, hello, thank you so much for being with us this morning. My name is Dr. Ridlin, and my bioinformatics class this semester has been working on a code project with Hudson Alpha where we have been analyzing different clinical variants. So these are genetic mutations that are found in patients, and they are of unknown significance. So it's comparing kind of these mutations to the normal wild-type genome and trying to infer some of the changes that occur. This is Sam Menard, and he has been working on a particular variant that he is going to be introducing to you. And Sam is one of our fantastic biology majors in our department. All right, you ready?
Yes, ma'am.
All right.
How are you all doing today? Like she said, my name is Samuel Menard, and today I'm going to be going over the variant of unknown significance of the V71L mutation in autoimmune role of CTLA4. So what is CTLA4? CTLA4, or cytotoxic T lymphocyte-associated protein 4, is located on the chromosome 2 on the Q arm band at location 33.2. It is a protein receptor that functions as an immune response checkpoint or regulator of the immune system and downregulates immune responses. So this is the P arm, or yeah, the short arm is the P arm and the long arm is the Q arm, so that would be located about down here. And so now you're probably thinking like, Sam, what is that? Basically, the gene codes protein and stops the overfunction of T cells that multiply rapidly if they're not controlled. So it downregulates, and downregulating is basically lowering the rate of a process, so it just lessens the receptors per cell. So the pathological phenotype of this. So mutations in the CTLA4 gene have been linked with insulin-dependent diabetes mellitus, so DM1, Graves' disease, which is what you see over here. That is what happens if Graves' disease goes untreated for a very long time. Hashimoto thyroiditis, celiac disease, SLE, thyroid-associated orbitopathy, and other autoimmune diseases. So DM1 is, like I said, that's just diabetes. It is a multifactorial disorder of glucose homeostasis that is characterized by susceptibility to ketoacidosis in absence of insulin therapy. So that shows as polydipsia, polyphagia, polyuria from hyperglycemia-induced osmotic diuresis and secondary thirst. So my variant, a point mutation indicates that a single nucleotide has been changed within a sequence and therefore potentially changed the entire codon. In some cases of a point mutation, so like GCC to GCG, that's alanine to alanine. There's no change in the codon that is coded, therefore there's no phenotypic change in expression. A missense mutation is a point mutation where a nucleotide changes to, a nucleotide change leads to a codon change that codes for a different amino acid. So at this location, 71 in the CTLA4 gene, a missense point mutation causes the first guanine nucleotide to be replaced by a thymine nucleotide, so GTG to TTG. This changes the entire amino acid from valine to leucine. Then over here is an impact chart of all of the unknown. There's also the one benign that is confirmed, and 3 pathogenics that have already been confirmed. And then here's mine up here at location 71, and it has a CAD score of 3.7. And that CAD score just kind of indicates like how deleterious a mutation specifically is. Now, this mutation has incomplete penetration, so just because you may have the genotype of it does not mean that it will phenotypically express within your body. So you're not guaranteed to get these diseases that are very damaging to you. This is an image that was printed through Yasara, which is used to show the 3D structure of different proteins and molecules and things of that nature. This is what the wild type looks like, and then this is what the variant looks like after the mutation occurs. And you'll notice that there are two identical sides, and that's because there's a molecule A and a molecule B. And this is what they look like up close. So this is on molecule A before the mutation occurs, that valine is there. And then this is also on molecule A after the mutation occurs, and you'll see that side chain difference up here. And then at the bottom in the green, that's the molecule B. This is before the mutation occurs, and then this is after the mutation occurs. And once again, you'll see a big difference in the side chain. So the difference, like I said, you go from guanine to thymine. This one over here is what a guanine looks like, and this is what a thymine looks like. The main difference between them is that guanine has two rings, it's insoluble in water, and pairs with cytosol. Thymine is exclusive to DNA, so it can be translated into or from RNA, and it pairs with adenine. And then this is the valine molecule, and then this is leucine. So you'll see this huge side chain addition to that here, highlighted in red. And then the variation location in the protein. So there are three main domains. There's the extracellular, which are positions 36 to 161, and then the helical domain, which is positions 162 to 182, and then the cytoplasmic, which is positions 183 to 223. Since my variant is at position 71, that means it is therefore extracellular. These are the polyphen 2 results. Polyphen is used to, it's a huge program that goes through all kinds of math to determine how damaging a mutation is or what it may do. And mine has come out to have a polyphen score of 0.975 and 0.804. That means that it's probably damaging. The difference between these two scores, though, like you'll notice over here, there's HumDiv and then HumVar. The difference is that on HumDiv, it goes through the Mendelian disease variants versus the divergence from close mammalian homologs of human protein. So basically, us plus other species that we're close to or that our genome is close to. And then HumVar is just human variants associated with the disease, except for like cancerous mutations or loss of activity in human polymorphisms with no reported correlation to disease or other effect. And then also in polyphen, you'll notice that there's a multiple sequence alignment chart. This is basically just showing that throughout all of the different species, there's a valine located there. There's a valine amino acid located at that position on this particular gene. And then I also ran this through SIFT, which is a program that tells you whether or not your body would find this tolerant or intolerant. And the results were kind of conflicting, and it showed that it would mostly be tolerated by your body, but I didn't particularly include that because that was, like I said, conflicting with the results that I had found. And then these are my references.
Questions for our speaker?
So I'll start. So Sam, I know you want to do genetics for your career. What about this project excited you the most?
Getting to know how many different programs there were to kind of explore this world, and then also like Dr. Triplett was my genetics teacher, and he did a really good job. I really enjoyed his class. He made genetics fun to learn. But for me, I like to do things a lot more hands-on, and this project allowed me to actually go through these programs and learn how to use these programs and kind of explore a little bit more and research on my own. Yes, sir.
Autoimmune diseases in my family. My grandfather and my cousin both have lupus.
Yes, sir.
So is this something that you would encourage people who have that autoimmune disease in their family history to sort of look for?
Yes, because it does affect like your autoimmune response, but this would not be something for you specifically to research if you're not in that field. However, if you do choose to, basically if you just want to know more about what's wrong with you or what you feel like is wrong with you, then yes, you can look into it if you'd like. Anyone else? Yes, ma'am.
A question. So, like in recent
Yes, ma'am, actually. There was a study done by the CDC that showed linkage between these two. So potentially not confirmed yet. Anyone else?
All right, let's thank our speaker again.